thank you to all to everyone who's in attendance today. And again, thanks to our instructors who are going to be, who have been and will be teaching this workshop series. This is the third day out of 4 for talking about genome to phenome, specifically for non-biologists. As part of the AG2PI initiative, there are several other events, and I do want to just let folks know that there is going to be a field day, which is basically a series of talks about the US-based aquaculture field. We've got 3 speakers for that, and that will be held April 21st. If you're on the AG2PI email list, you will have gotten notifications for this. If not, you can find this on the AG2PI website, which is AG2PI, the number 2, .org under field days. And so today we're going to have 2 talks. The first one will be on modifications of genomes and transcriptomes through something that we call the epigenome and the Epi Transcriptome. This will be given by Dr. Chris Tuggle at the Iowa State University. And after Chris, we're going to have a talk by Dr. Ryan Barthelme at the University of Arizona, talking about biological complexity and systems biology. We've done a lot in terms of the naming of parts, but ultimately when it comes to understanding how genotype to phenotype works, it's about how these components come together. And we're often talking about thousands, hundreds of thousands of them inside of a cell to give rise to these emergent properties, often what we call behavior or phenotypes of these organisms. And so with that, it's my great pleasure to hand it over to you, Chris.
Thanks, Eric. I'm going to just show my screen here.
And as you're doing that, Chris, I just want to remind both you and Ryan, if you haven't done so already, just make sure the voice to text, which you have up, Chris, is functional. Thank you.
Yeah, I can see it, so it should be good. Hello, everyone. As Eric already introduced, the session 5 is on epigenetics and epitranscriptomics. And so I'll just, what I'm going to try to do is take what you've heard of before on gene expression and transcription and translation, some of the core initial processes, and now I'd like to add to you or give you some information about how those processes are regulated during life, and focusing primarily on epigenetics and that area for the 30 minutes or so that I have. So epi is above or upon, and so epigenetics is a study of heritable phenotypic variation that doesn't involve changes to the DNA sequence. An important component of that is that it's reversible. So reversible molecular changes. This is not, again, this is not at the level of the DNA sequence, but it affects biological function, and that's often due to environment. And so the study of epigenetics is really how the genome functions and how it functions across different levels of organization, cells, tissues, developmental states, and environments. And I like to think of it as the mechanism for how our cells use our genomic information to respond to stimuli, to interact, to adapt, and to grow. So this is clearly a very large area of scientific inquiry, and I'm trying to get you through it in about 30 minutes. So a first concrete example for this whole large area of an example of epigenetics effects on phenotype. On the left, you have a queen bee. On the right, you have a worker bee. Now, if you know something about bees, you know that they're all clonal, and these two from the same colony essentially have the same genes, same gene sequences. But they're different. The queen bee has certain behaviors that are very different from the worker bee, and she can lay eggs. The worker bee can't. So there's a lot of physiological and behavioral differences. But how are they different? What caused them to be different? Well, it was a different food that caused this huge phenotypic difference. And so we'll answer that question later, but you might want to think about that. How could a food create such a phenotypic difference? So the main thrust of this discussion today is that we're interested in selective gene expression, because at least for me, it defines function. Our bodies are a bunch of different parts, and so Eric, I'll be adding lots and lots of parts and lots of names again today. So it's strictly biology, I guess, not into systems. But those body parts are built from different kinds of tissues that are made up of lots of different kinds of cell types. But each cell type or tissue is expressing different sets of genes from the same genome. So I like to think of a useful definition of different or specific cell types or tissues as the pattern of genes that are active in those cells in that tissue. So in fact, that's primarily what I do when I'm trying to understand what's happening, is to try to identify what's active in that cell or tissue. So how are cells selectively expressed? We've covered central dogma and a bit about how genes in general are expressed. We talk about transcription and translation, but how does selective gene expression work? Why are some genes on and some genes off? How can we measure it? How do we use it to define cell types, especially across lots of different conditions? And then even the interaction of all these. Because what we really want to do is take a genome and predict phenome, right? So we want to be able to predict phenotype under different conditions. So we have the same gene, same G, but different P. So this is a problem or an opportunity, depending on where you look at it. So we can look at growth, we can look at maternal or other kinds of diet changes, or even something like interaction between environment and genotype. So this is an example from a textbook on genetics where you might have two different genotypes that are different height in plants under dry conditions, but potentially under a different condition, like a wet condition, they switch, and the one that was doing poorly under one environment can do much better under a different environment. So this is an example of a genetic interaction. My focus today really is on transcription and what controls transcription. So the two things that I would like to cover is where and when a gene is transcribed into RNA, and that's the major level really controlling the activity and the expression of genes. There's obviously a lot of regulatory control over how much protein is made and where that protein is put in the cell or in the tissue, but a large portion of the differences between cell types and between tissues is the RNAs that are expressed. So that's where I'm going to focus. I'm also going to focus on the compactness of the chromatin, and I'll talk about that. And Cliff discussed a little bit about that, but really to try to get into how we measure openness and the nature of the chromatin through a number of different assays. So what are the molecular mechanisms that alter chromatin structure? Well, there's three major ones: changes in patterns of DNA methylation, modifications of the protein component of chromatin, so modifications to those histone proteins that you may have heard about. And then I won't have much time to talk about this, but there are also RNA molecules that affect chromatin structure and gene expression. And those can range from small RNAs to very large RNAs that can bind and silence chromosomes like the X chromosome. So a little bit back, taking a step back, if we think about the first principles of gene expression, a gene is often drawn this way. You talk about exons and introns, and maybe you talk about a transcriptional start site and an end. But what's missing? What's missing is what I want to talk about today, which is regulatory elements. We normally think about two major kinds of regulatory elements that are at a distance, and they could either be completely different gene, like something that might express a transcription factor, and we call that a trans component, or something that could be as much as a megabase away of DNA sequence called an enhancer, something that acts on a particular target gene. And it acts on that by binding a transcription factor that interacts with a core promoter complex, often including the RNA polymerase complex. And there's a short animation that I'm going to show here, and hopefully I can get this to work. Okay, Can you see that? That's biological animations. And I would recommend, I think Cliff has shown one of these videos. The DNA Learning Center has a number of different animations that are kind of cool to watch. The one I want to focus on is transcription. And I'm going to turn this completely off so there's no feedback or anything. To set this up, what I would like to show you is they're going to depict the best current resolution and current view of what a promoter and the transcriptional regulatory transcriptional complex looks like at the beginning of a gene. And so that's what they're going to start with here. And then what they'll also show you is a depiction of an enhancer region with a protein bound to it coming in and starting the initiation of transcription. And then they show a little bit of RNA transcription. So this will take about a minute, and I just wanted to give you a view of this. So essentially, they're showing you that a lot of times there's a promoter for a gene, and the gene goes off to the left here. This core promoter complex has a few proteins, but nothing that are bound to it, but nothing is being transcribed yet. But they can identify the beginning of the gene. And then what will happen is additional proteins will be recognized that they need to come in and bind. And so then you create a latent and a protein complex that's ready to start making RNA. And then what happens and comes in very quickly here, as you'll see, is a protein complex is looping in on the DNA, and you start transcription. And so then the RNA polymerase is now making the RNA. And this is in actual what they think of as real time. So the RNA molecule is being created very, very quickly as the polymerase moves down the DNA. So that was the enhancer coming in and interacting with the promoter. So a little bit more on first principles. This selective gene expression model has been elaborated over many years. And here's a little bit more complicated view of it. We're describing perhaps two different genes, that one is expressed in the brain and another gene that might be expressed in the limbs. And so you have brain factors that are binding an enhancer and then are turning on gene expression just in the brain, or for example, gene expression just in the limbs. And what's missing from this view is really thinking about the logic of this. There's really something missing, and you might think about what that is. But the hint here is a pile of turtles, and maybe some of the statistical geneticists and logic people might thinking a little bit about the view of it's turtles all the way down, if you've ever heard that. Essentially, that's a funny story behind the infinite regression model. And let me just set that up for you a little bit. The logic model is that if we can explain tissue-specific expression by a brain-specific enhancer binding to a gene, and then that turns on expression in the brain, well, that doesn't really solve the problem, the logic problem, right? Because that just puts it back one step. How do we have a transcription factor? How do we explain regulation when the transcription factor itself has to be specifically expressed? In other words, the brain-specific transcription factor has to have a brain-specific transcription factor binding to its enhancer so that it can be expressed in the brain only. And then that factor has to have a brain-specific factor, and that factor has to have a brain-specific factor, and on infinitely. So this is what we call the combinatorial model of gene regulation. That explains how we don't have turtles all the way down. And if the old Hindu mythology is that the flat Earth is on the back of a turtle, and then somebody asked, well, what is that turtle standing on? And then the person said, oh, it's turtles all the way down. So the idea here is that we have to have this, what solves this infinite regression problem? And what solves it is this idea of combinatorial models of gene regulation. And so the idea is that transcription factors combine together to make new selective expression patterns. You don't have to have a specific transcription factor. You just can need combinations of them. And one of the best examples is the model for gene regulation in plants in floral organ identity. So different parts of a plant flower have different identities, and you need only 3 factors to describe and specify each of those factors, because you can have A factor all by itself, you can have A and B together, and that makes a new organ. You can have B and C together, and that makes a new organ. You can also have C alone, and you can have even more variable inputs and outputs by just, in fact, having something beyond on and off. So you could have a gradation of A and a gradation of B and so forth. So that's the story behind the turtles all the way down. So you might have the right transcription factor present in the cell, but it doesn't do any good unless the parts of the genome are accessible to be bound by that factor. So it's also very important to have the genome be accessible. And so how do we recognize accessible regions of the genome, and how do we look at active versus inactive enhancers? And so the Human Genome Project was started more than 10 years ago to try to answer that question for the human genome. And then plants and animals have followed, starting up only in the last few years. So they spent about $150 million to try to answer what part of the genome is active. And so, of course, they're looking at RNA levels and what part controls this expression. And so there's a lot of different assays that look at biochemical structure. And so that's what a biochemical structure of the assays of chromatin structure. So just briefly, how do you measure RNA? You might know this already. You start with RNA, you convert it to a cDNA product, and then you can clone and amplify those fragments and then map them to the genome until you get peaks of RNA reads that are localized to a particular place in the genome, and then you know that that's the place where the RNA came from. And so that's just showing here. And this is an example of all the reads that pile up into peaks at each of the exons for the GAPDH gene. And so these are the RNA-seq reads, and you can see that this would be the way that you could actually identify what parts of the genome are expressed into exons and what parts are not. You can also think about measuring chromatin accessibility, as I mentioned before, what parts are open and what parts are closed. And so regulatory factors need this region that's to be free of any nucleosomes. And there's a couple of different ways that we can recognize these regions that can be bound by factors. One is DNase I hypersensitivity, and that's a really complicated assay. It's very difficult for people to do. So what's come around is this new assay called ATAC-seq. And essentially, it's very similar. The idea is that a TN5 transposon or transposase can cleave anything that it can get to. So anything that's open enough that for the enzyme to come in, you can recognize and you can see it'll be digested out of the chromosome. And then the TN5 actually puts in sequencing primers, and so that's very convenient. It allows us to basically recognize what parts are open and which parts are closed, because we can clone out and sequence the open regions. So we can map those to the genome, we can define the peaks and so forth. The other ways that we look at chromatin modifications is the histone modifications, which I talked about before. There are more than 100 different post-translational modifications of those histone proteins, and they include a number of different things. We'll focus mostly on methylation and acetylation today. One of the major ways that we look at what parts of the chromatin are modified is use an assay called the chromatin immunoprecipitation assay. And the basic idea behind that is that to recognize what parts of the genome are bound by factors or bound by a particular modified histone is to cross-link, to fragment the chromatin. And then if you add an antibody and use an antibody immunoprecipitation assay, that will allow you to identify those places that were closest to those histone marks or to a transcription factor. And it's immuno because an antibody is used. But essentially, you end up with something very similar to the RNA-seq I showed you, where you pile up a whole bunch of reads from a particular region of the genome that was enriched for sequences in the library that you have. And your readout is really coverage by those regions of sequence that was either bound by a factor or had a histone bound to histone modification close to it. And so putting it all together, modifications at the histones and chromatin accessibility you can take those results and those peaks, as I mentioned before, you can put that kind of information together to try to predict functions of genome regions and to really take chromatin accessibility and these histone marks to validate these regions. So for example, there are some histone marks that are known to always be near active enhancers, or those that are near active promoters. And you can then use that information to predict different states of the chromosome as you go down the chromosome. And this is an example of data from one of our projects, the collaborator on a pig fang project, where you've already seen this, this GAPDH RNA-seq data, but all of these data are from different histone marks or other transcription factors. And you can see that the sequences pile up in the region near the promoter of the GAPDH gene, because the gene is transcribed from right to left. But there's also another gene here that also has marks. And so this, you can see just visually that we're able to identify histone modifications or open chromatin and then predict what is the function of these chromosome regions for that particular cell type or tissue. And it'll be different for a different tissue. This particular tissue is spleen, but we'd see a different pattern in this region if it was a different tissue. And again, putting it all together, we can actually use a algorithm to predict chromatin states. And I won't go through this in detail, but you can pick out, this is known, these patterns are known based on having data of particular enhancers and promoters. And you can see, for example, if we look at the pattern shown in number 4, so the fourth chromatin state, that's predicting an active promoter because it has these 2 marks. And if you look to see where those marks are relative to all positions in the genome around the transcriptional start site, you can see that the signal is very, very focused right on the transcriptional start site. So that marks an active promoter. And you could do the same thing with other combinations of marks. So that's histone modification and chromatin accessibility. The DNA itself is also modified, and the primary modification is DNA methylation at a cytosine to make 5-methylcytosine. And this is important because transcription factor binding motifs are recognized by those transcription factors often in the groove of DNA, and the methyl groups actually can prevent the transcription factors from recognizing those sequences. And so they tend to be preventive of DNA being bound by protein, and so that tends to be a negative signal. The way it's assayed is converting unmethylated Cs to use uracils using a particular chemical, sodium bisulfite. The methylated C is protected from that treatment. So then afterward, you can see the sequences that have changed and identify those Cs that are methylated and those Cs that are not. Methylation, as I mentioned, it's often a repressive signal because it's preventing transcription factors from binding or other factors. And so you normally think of methylation as a negative signal. And it can be regulated. You can put methyl groups on and you can take methyl groups off. Methylation is occurring through an enzyme called DNA methyltransferase, for example. So if we go back to our example of the differences between these 2 bees, how does food create a queen? What is the epigenetic effect? And really what it turns out is that queens are fed a food called royal jelly. And when they analyzed this royal jelly, they found that the substances in the royal jelly actually turn off a DNA methyltransferase. So they're decreasing the amount of methylation. And so royal jelly decreases that methylation, so more genes are expressed. So methylation is a suppressive effect or an inhibitory mark. And so you're suppressing the inhibition, so you're having more genes expressed. So this epigenetic effect is actually turning on genes that can create a queen. I won't go into much detail here because we're running out of time, but I wanted to just briefly mention that there are many other aspects of epigenetics, and one of them is imprinting. Imprinting was identified in mammals because you can't create a viable embryo from 2 female-derived genomes or 2 male-derived genomes. You have to create an embryo from one from an egg and one from a sperm. And that's even true for inbred mice. So we know that where the genomes are identical, so it has to be an epigenetic effect. And one theory for this, why would you have this imprinting, is that the mother is trying to limit the use of maternal resources, whereas on the paternal side, the male side would just like to have big, fast offspring. But the mother, of course, has to limit her use, the use of her resources for the baby. And in fact, that's been borne out by which genes are functionally affected by methylation. But the major mechanism for this is DNA methylation. There's also methylation and genomic imprinting in plants, which I unfortunately don't have time to go through today. But if you're interested in plants and looking at imprinting in plants, there is quite a bit of research being done in that area. And I won't go through this, but there are many different epigenetic effects on phenotype that have been documented. The one thing I wanted to just briefly mention is what's called transgenerational epigenetic effects. This was recognized that there was a relationship between food shortages in parents and obesity in the offspring. And they came up, and this was true for looking at records in Sweden in the farms where there was records of food shortages and excesses. And so they came up with this idea of a thrifty phenotype hypothesis. And the idea is that if you have food deprivation in utero, the fetus gets very, very protective of calories. And if you then afterward have a very caloric rich environment, then the offspring that's efficiently storing excess calories can become obese. And they see this also in a Dutch famine, where they saw the same problem. Pregnant women that were starved during World War II had children of those women, had increased metabolic and cardiovascular disease. And they was really interesting, as they found that also translated even to the grandchildren of those women who were starved in the mid-40s. And the mechanism for this has been shown to be at least involve DNA methylation. And there's many possible mechanisms, but one of the major ones they believe is DNA methylation. And a lot of work has been done on this, a very large field of study looking at transgenerational epigenetic effects. And finally, a couple of slides on Epi Transcriptomics. RNA can be epigenetically modified as well. So this is post-transcriptional, and then you have modification to the RNA. This is not changing the RNA sequence, but just modifying the bases that are there. And there are over 140 different modifications that are known. There's a lot of different aspects of this, but why would we want to modify the RNA? Well, it can change which proteins are binding, so that changes the activity or the phenotype of that RNA, how stable it is, whether it can be folded correctly, and whether it can be translated efficiently. So this epigenetic modifications can then affect RNA function and therefore phenotype. There's many projects in plants and in animals that are taking the ENCODE type of project forward and really trying to understand functional genomics in the context of our agriculturally important species. And if you're interested, there are resources that I'll show there. You can look at those. There's a very large field of study on epigenetics. And as I just wanted to remind myself, I wasn't really able to talk about RNA interference at all today. This is just a summary, which again, I won't go through because we've run out of time, but it's there for your reference. And so I guess at this point, I'm done, run out of time. And here are the discussion questions that maybe we can discuss a little bit in small group. And for that, and that's where I'll stop. Thanks. So would anybody like to start with answers to question 1? Breakout room 1, maybe we start with you. Looks like you've got an answer up there. Different transcription factors combined together to give different expression patterns. Yeah, that's right. I think the answers here are pretty straightforward. And that was what I was trying to get. It looks like everyone really got the main point. Maybe we could go on to 2. So breakout room 1, that's good. So I think all the answers there are pretty straightforward, and the question was that way. So I think we could maybe even skip question 2 if you don't mind. We'll go on to maybe the ones that we discussed in our group, and maybe some of the other groups did as well. It says, what's the basis for genomic imprinting? Why does it occur? Maybe we should have somebody from breakout room 1 talk about their answer. I know who was in breakout room 1.
See,
I get somebody to talk, right?
Yeah, I was, but I think
Anthony?
Shelley came up with the answer.
I'm not sure Shelley's on. Maybe you want to go ahead.
Okay, so we thought that one of the parents can turn off expression of certain genes, and that way one of the parents will dominate characteristics of the progeny, I guess. And then why does this happen to regulate normal development or growth, things like that?
Yep, that's great answer. In fact, that's really correct. The good example I have is that the paternal side activates the insulin-like growth factor genes, and so to try to make the fetus grow fast, because it doesn't care about whether the maternal resources are used up too much or not, just wants a healthy big baby. So exactly, and then the parents, the maternal side will do something different, so turn off genes. Breakout room 3, cytoplasmic signals transfer to gametes to maintain epigenetic marks. Anyone other than the organizers have something to say from breakout room 3? In addition to what's there? So what about breakout room 2? You've got parental genes are one of them that can activate or be microdeleted, I guess. You guys want to discuss that? What do you think about microdeletion?
I can speak about that. I know one diseases in humans. It can cause a disease, I can say. It's because of microdeletion of some genes, parental genes. One of them can microdelete it, and these genes not expressed. That's why these diseases can occur.
So it's causing a change in genomic imprinting or something along those lines? Well, that's interesting.
Its name is uniparental disomy.
Okay.
It's kind of thing.
Oh, that's interesting.
Very
good. Anything else that people want to, offer or talk about for genomic imprinting? Okay, let's talk about question 4. So I'm not sure we've had anyone talk from breakout room 1. Can you guys talk a little bit about how your grandfather's diet can affect your health?
So I guess I'll.
Anthony, you talked before, I guess, right? Didn't you? Yeah.
Yeah, I can wait for someone else, but.
Well, no, go ahead. That's fine.
So we thought it's two layers of progeny, but the diet of your grandfather's diet can cause epigenetic changes in your parents, and then it can stay on to your generation.
Yeah, and I switched it a little bit because I had talked about the two examples I gave were basically grandmother's side, right? So something happening in the uterus. So what do you think about the grandfather side? Do you think that could happen? Could have modification of the grandfather's diet to change the offspring?
That's a good question. I don't know. I'm guessing, yeah.
Yeah, there's some evidence for it. It's not completely clear whether it's actually when the grandfather was in utero, and so his genotype, his phenotype was changed in utero by epigenetic mechanisms, and then that passes down. But there is some evidence that it can come from the paternal side as well, and it's not completely clear how. There's a little bit more of a broader, it was sort of a thought question.
Okay.
And we had somebody talking about hard candy and diet soda. I don't know if you want to sort of extrapolate on that a little bit more, but
I do want to hear about that.
yeah. I don't know if this is family history time or apparently not. So it looks like I think the other answers are great. And maybe we should just go to question 5. Is there anyone from breakout room 2 or 4 that would like to talk about their answers to question 5?
Yeah, I can talk about this one. This is Chris Hostetler. So heat and or lack of rain are environmental effects that change gene methylation in seeds, and then thereby they turn on or turn off genes in the next generation of plant. I don't know if that's how it works in plants. I'm an animal guy, but I mean, theoretically, I guess it would work very similar to the concept about transgenerational epigenetics in animals, right? So,
Very good.
don't know if it's right or not.
I think it's a good answer, and I don't know what the mechanism might be in plants. I don't know if there's some evidence that the gametes are changed during the plant's life to respond to those environmental changes, and then the seeds are then different, or if it's something that happens primarily within the seed. I'm not sure.
Yeah, I don't know that either. I would. Oh, I'm sorry, Cliff's answering.
In plants is that gametes don't get made until the end of development.
Exactly.
Yeah.
And there's no uterus, right?
Right, and you don't. All that meiosis is at the very tail end of development after flowering.
Yeah, interesting.
But it's always a biological process, right? You can modify it at multiple stages. Any other answers? Breakout room 2. You indicated DNA methylases and methylases and histone modifying enzymes could respond to the environment. I think that's a great answer. Certainly, if we modify the modifiers, we could certainly change the epigenetic patterns.
So Chris, that makes me, Cliff's comment makes me wonder about, the seeds being produced at the end of the life of the plant. That makes me wonder about the plant's ability to conserve, right? So in animals, we talk about a nutritional impingement on the mother, and she will sacrifice her own tissues in order to support the pregnancy to a certain extent, right? So do plants have that capability too? If they see all of a sudden a drought in late stages, could that be the trigger then that would change seed formation and DNA formation within the seed? So it might be stage specific drought and environmental effects. So anyways,
Yeah,
just thinking about
that's
that.
That's good insight. Yeah, I don't know if other people have a comment. I mean, definitely the starvation phenomena, the the nutritional phenomena that we see in humans is definitely stage specific as well.
Yeah, exactly.
There are a bunch of plants that won't really flower until the conditions are conducive to the seed, right?
Yeah.
So, you know, out there in the desert, you guys are
Yeah.
you know, where Eric is. That's pretty much, and Ryan, that's pretty much the story. That a plant will survive and survive and survive in hideous conditions, and then as soon as it rains, bang, everything flowers and makes seeds and disposes of them everywhere. So the kind of conserving things to give them to your offspring works that way in plants.
Great. Okay, I think we're getting close to the hour. Are there any last questions that people might have, or other insights?
I might have a question, just a quick one,
Sure.
if that's okay. So is there any knowledge behind how costly these modifications are? And in the context, I mean, is it easier for an organism to modify its DNA, or is it easier for it to modify proteins and RNA and keep them in cytoplasm?
That's a great question. I don't know that anyone's attempted to try to, estimate, sort of the metabolic cost for modifications, period, let alone, one type versus another. But it's an interesting question.
Yeah, and I'll take just a quick like stab slash guess at it. So when it comes to the modification of these different elements in a cell, genomes don't do something called turnover. We talk about turnover in terms of taking an RNA molecule, breaking it down, and then rebuilding it. Overall, that's relatively energy expensive in comparison to putting down a modification. These modifications tend to be very small biochemicals, usually like a methyl group or something along those lines, and there tends to be relatively easy and inexpensive chemistry to do that. But tearing apart an entire molecule and then rebuilding it, that's where a lot of the cellular energy goes. So if you're putting marks down on the genome, they basically stay there, but there is additional machinery that's around to what we call maintain it. It goes through, it checks it, it puts them down, or it removes them, and that's constantly in action. And same thing is happening with proteins and RNAs. When those get methylated, or sorry, when those get modified, some of those modifications are specific to target them to degradation. So if you're targeting them for degradation and then immediately you need to reuse them again, you'll build them back up. And so I imagine there's just more energy that's being spent in that process. I'll stop my guess at that point.
Great. Well, we're right at the hour. Do we want to take a couple-minute break before we start the next session, or do we want to just go straight in?
Yeah, no, I'd say take a quick 5 just so people can take care of business if they need to. And how about we meet back here at 5 past the hour?
Okay, let's do that.
Perfect. And with that, since we're past our time, it's my great pleasure to introduce again Ryan Barthelme. Ryan, I'm going to hand it over to you.
Thanks, Eric. So I get to talk about one of my favorite topics in biological research, the systems biology and networks, something I've been working on and thinking about for a long time now. And so I'd like to start off with just this piece of art, 1970s sci-fi art. So like, why would we even want to consider things from a systems biology perspective, or what do these networks even mean in this context? So I chose this sort of space colony looking piece of art because ultimately this systems level knowledge is important for something as extravagantly sci-fi as space colonization and travel. And I think that it's a good sort of grounding point of how little we really know. Like, we don't actually know enough about how biological systems are connected to each other to be able to pull off a floating greenhouse like this in outer space. I mean, we can do minor hydroponic experiments on the International Space Station, but we're really still just barely scratching the surface of these topics. And so when we consider what exactly is a biological system, we're talking about a mathematical space that we want to consider these biological processes and interactions between biological entities within. And these systems can vary in scope depending on what questions we're asking and what level of all of the different myriad of ways we've talked about observing molecular biology in genetics and phenotypes. And then specifically for going from genomics to phenotypes, the system is going to act to constrain the phenotypes through molecular and environmental interactions. So this is a great lead-in after talking about all of the epiomics modifications, how the environment exerts some level of control over how genes are expressed or how they are stored inside of a genome. We could think about these biological systems at different scales. So we can consider a single cell, single cell. It could be a bacterial cell where that constitutes a whole organism, or it could be a cell that is part of a much larger organism like a cow or a human or a plant. And inside these cells, we have everything that we've talked about so far in this series. We have this genomic information. We have this gene expression occurring, and we have metabolic interactions occurring from outside and within the cell itself. And then when we consider the cells as part of a multicellular organism, we're talking about many, many cells with many different metabolic pathways and interactions, like what Chris was discussing about the brain and the feedback loops that we have inside of these different organisms. We zoom out even further, we can think about it in terms of an ecosystem. We think about the totality of environments and organisms in a given area in the same place. There's a large number of metabolic interactions between organisms. So if we think about this in terms of like your production and consumption of biomass models that you may have had in the past in ecology classes or in introductory biology, and all of these systems are going to have spatial and temporal relationships. I believe it was Cliff that said that cells are crowded, busy, four-dimensional places. So these reactions are occurring in different organelles within eukaryotic cells, and those are happening on different time scales. They're moving in between the organelles into different spaces. But how do we put this in terms of a network? So like we need to establish what exactly a network is. So these are two diagrams of different kinds of networks. These aren't all of the kinds of networks that you could possibly have, but this is just a good overview of what networks are and what we can do with them. So right here, we have the directed network graph. You can see there's arrows pointing to between the nodes or vertices, the blue points on the network. These are linked together by edges or these are the connections between the nodes. And you see in the directed network, there's an arrowhead that points in a particular direction. And you see the undirected network graph, there's no directionality associated with it. And we can also have things that are in between, where part of it is undirected and part of it is directed. Call those hybrid networks. So network graphs can have any of these directional or undirected edges. And when we talk about biology, we're often talking about scale-free networks, which have like a hub node with many edges going to many different nodes. So you could think of the hub node as like potentially like the organism of interest. So if you have, say, a collection of genotypes that you're interested in looking at the phenotypic measures, the genomes and the genotypes of an organism might form that hub node of that scale-free network, and then expand out to include a lot of the different molecular interactions and assays and methods that we've talked about thus far. in this AG2PI workshop. So what we do when we're doing a network analysis, there's sort of a basic recipe of how we would start to do this. So this is an example over here of what I was just talking about with this scale-free network. This is an undirected scale-free network with the hub node in red. But before we can even get to this point, we need to clean up our data. So biological data is very messy. Often it includes some categorical variables that we can't necessarily include in some of these algorithms that require continuous or binary data. So we have to do some cleaning and transformation before we can get to step 2, which is learning the structure. So this learning of the structure is what generates these graphs on the side that you see on the right-hand side of my screen. And some of these methods will allow us to impart some of our knowledge into this process and generate these directed, undirected, or hybrid graphs. But that's just telling us the relationship. It's not really doing anything quantitative yet. So the next step we would need to do is to fit the data to a graph. There's a whole bunch of different algorithms out there that do this, and that's well beyond the scope of this workshop. And then the next step after that, same thing. It's very complicated. There's a lot of algorithms associated with this validation and evaluation of fitting the data to the graph. And then ultimately at the end, where we want to have some predictions or associative power. And this can come in whatever statistical flavor. I don't want to pick sides in the deterministic versus Bayesian battle that has been waged in statistics for a while. But you can go in both directions with those associations, and you can also think about it and potentially in some instances in terms of causality, right? So that's something we're interested in, what is causing us to observe these phenotypes. So ultimately, at the end of the day, we're kind of interested in causality, but we're not quite there yet, scientifically speaking. When we talk about computational versus biological networks, really what we're talking about is kind of what the edges represent. So in a computational network, these edges are going to represent the connections or the different computations that are occurring between data that reside at the nodes or vertices of the graph. And in a biological network, what we're talking about with the edges is the interactions or causal relationships between the different observations that we have. And these are dense with many directions. And like I said earlier, they're often scale free. This makes it a very, very difficult problem. If you're a computer scientist, you might have heard of the NP-hard problem. This is exactly what that is. This isn't like, there are some famous graph problems like the traveling salesman problem, where how do you have a traveling salesman go from city to city and not overlap with the routes more than once? So this is even harder than optimizing that when we're talking about biological systems. They're so complex. And we could think about it in terms of different scales, and these are all happening on different time scales and different biological scales. This isn't a very new idea. Sewell Wright came up with this in 1920. That's over 100 years ago. So he's probably most famous in statistics for being R.A. Fisher's antagonist through that entire time period. But he was also starting to try and develop networks before we even had the computational capacity to do it. And he was doing this by mapping phenotypes of guinea pigs for the U.S. Department of Agriculture. We talk about these scales, it's a very continuous sort of level. So this is a diagram from a research project that I'm on that we developed. And we're talking about you can see a lot of the things that we've discussed right now in the AG2PI workshop series have continued and continue to grow. And we go from organelles to cells to tissues to the anatomical systems to individual organisms and then populations of organisms that all exist in this continuum of environment and time. So if you remember from Cliff's talk, that we're thinking about this in terms of we're starting with the DNA as the instructions that encode, and we're going outward from there. So we could do network analyses on single cells. So this is an example with, I believe, human subjects, where we're doing transcriptome sequencing. And if we pool the tissues together, we can look at potential interactions between the different tissue types. If we separate them into single cells, we can look at regulatory links inside of a single cell versus looking at how the system, i.e. a human being, their cells interact with each other. So there's these different modalities of thinking about this in terms of, do we want to pool the different tissue samples together to look at interactions, like to use Chris's example, between the brain and another organ in the body, or are we specifically interested in, say, like a hepatic cell out of a liver, and we want multiple hepatic cells from multiple livers, and we want to look at these co-regulatory links between those. But when we start to think about, so that's like, we're going from a single cell to multiple tissue samples in an organism to, okay, now we have our organism, but how then does that organism interact with this environment? And I like this figure a lot. Also a great lead in for the field day, the next field day, the aquaculture field day. So how are we going to put these crops and livestock into a broader context with their environment? So this is a great diagram from just Shriver and Vodstein, where they compare the environmental microbiota between cows and fish. So two very different environments. So the fish are constantly being inundated with their environmental microbiota. And if you're familiar with how fish physiology works, in order for fish to breathe, the water keeps passing across the gill epithelia, and there's a whole bunch of different interactions that can happen. And that's usually like one of the places of disease in fish. It's very different than cows, which graze on their environment. They might get up and move away from where they've defecated. So when we're starting to think about this in terms of how an organism is interacting with its environment, we're also considering how the environment constitutes a microbial community. And that'll be really important to think about. So we think about like a human gut. Inside the human gut, there are all these complex metabolic interactions going on between our microbiome in our gut in different places, which varies depending on where you are in the gut tissue, like what microorganisms are where. And it's all going to do these chemical transformations that the gut cells themselves can't do. So there's this network of a vast amount of microorganisms, millions and billions of microorganisms throughout your gut that are helping to facilitate the breakdown of these compounds. The microorganisms are getting a little bit of energy out of it, and you're getting the building blocks that you need to exist and thrive in this world. We can also think about these microbes in terms of markers. So if we have all of these different gut organisms that are associated with these chemical processes, if this is a paper that one of my former lab mates published, where they found these microbiome signatures. In this case, they call them oligotypes. So this is unique information associated with the gut microbiome of a cow, a chicken, a deer, cat, dog, swine, and human sewage. And so what this is looking at, this is showing us that there are unique microbiome signatures for deer, cow, chicken, cat, dogs, swine, and sewage. And so this is interesting when we consider this, when we consider that these are unique within an organism, but there's also shared environmental microbes across this. We can think about plants too. The same thing can be said. There's arbuscular mycorrhizal fungi that are helping get nutrients into a plant. And these are able to do things like recruit iron into a plant. And all of this bulk soil microbes are being recruited actively by the plant. And it's this interesting balance of the plant sharing metabolites to attract the microbes that it wants so that they can exclude the microbes that the plant roots do not want. And that will help the plant to avoid disease. So it's this constant turnover of these microorganisms interacting with the molecules produced by the plant and vice versa. So you can think about all of this, putting it all back together again. This diagram makes a little bit more sense in that context. So when we go from these single cells to tissues to anatomical systems, we also have these interactions with microorganisms which constitute other biological systems. And then that starts to scale in complexity from there. And so when we think about this, we're going to think about like the future directions of this. This is kind of where we're at the edge of the field. So this is where these associations get really complex. And if we go back to this image and we think about this in terms of how do we get from here to these system level interactions? How do we go from the genes and genomes all the way to populations? And then how is this all existing together in these environments? I think it's important that we start to think about agriculture in terms of ecosystem thinking. And this is an iterative process, and we'll need to include like new data, new methods in these network analyses as they become available. It's a very active research field in computer science, and the applications in biology are kind of coming in just behind the development of new algorithms. So it's an interesting place for us to be research-wise. I was working on a network analysis yesterday with some plant phenotyping, and I realized that the algorithm that I was using for the structure learning was published last year. So this is such a it's constantly evolving, this whole field of network analysis and how we want to put all of these organisms into this bigger context with each other and within themselves if we're talking about multicellular organisms. And so with that, I will take any questions before we go into the breakout room. So the first question, how do we put crops and livestock into the broader context with their environments? Breakout room 1.
We didn't get to that one.
Okay.
We went back.
What's that?
We went backwards.
Okay. So breakout room 2. So the relationships between crops and the environment relate the crop biomass to livestock growth to predict the efficiency of crop rotation. In hydroponics, there's balance between energy and nutrition. Does anybody from room 2 that's still here, would they like to expand on that?
Yeah, I'm from room 2 to explain the first idea between of the relationship between the crop and the environment. Because I study the weight. Usually weight can be used as the dual goal crop to feed the cow in the fall season and the early spring season before wheat jointing stage. So we can use the crop biomass to predict the cow growth before the during that period. And also we can combine with the environmental factor to make a prediction model to predict the crop production and also the livestock growth based on their data. That's my idea. Thanks.
Thank you. Breakout room 3. You need to consider interrelationships between plants and animals in the ecosystem. So question 2 is, what relationships can we derive at different biological scales that affect phenotyping? Room 1 felt it was a confusing question, and I apologize. It was purposefully meant to be a little confusing to spur on some conversation, because there are so many opportunities, as Room 1 pointed out, at different scales, at the molecular scale, at a cellular scale, at an organismal scale. All of these are going to eventually have some effect on phenotyping, and it's just a matter of figuring out exactly kind of what they hinted at the end, where populations of organisms can work together or work against each other in an environment. Breakout room 2 said that environment affects which genes are turned off and on at cellular level as well as at organism level. I did really like this answer from breakout room 3, where it says the measurement system will vary depending on the scale, from a microscope to a satellite. So I thought that was an interesting way of interpreting the question. And they specifically referenced like the nitrogen and carbon cycles, because it's interesting to think about the elemental cycling at those different scales. So if anybody has ever taken a class in biogeochemistry, you're very interested in how the different atoms that make up living things move through an ecosystem. So I thought that was a very interesting way of interpreting it. And then the last question, why might we want to pursue a systems biology approach? Would somebody from breakout room 1 like to expand on this? You had a pretty good answer.
I would love for my group or the group that I was in to respond, other than me or Cliff. Oh, I forgot everyone's name. Arthur will come to the rescue.
There, Selka was there, but did not say anything.
All right, well,
We're still here.
yeah, Arthur, do you want to say something, or?
Everybody is still on. Come on, folks.
Well, just for time, I'm happy to jump in there. But as we were thinking about this question, you know, one of the key things about these relationships is really understanding how, you know, inputs can be affecting an organism in and of itself, but also all of these knock-on effects that it could have with the environment. So we thought a little bit about. There was a great example the group brought up about how fertilizers are often added in agricultural settings to help plants grow and make food. But there is this negative impact that it could have on the surrounding environment. And that it's really important to always be thinking about the bigger picture, the idea of these emergent behaviors that can happen as you start to do this. Space junk being another example, which is it's great to have a bunch of satellites up there giving us internet or communications, but you know, in the future they can lead to space junk. So systems biology is really there to help paint that big picture. When we take a look at all these individual components, what does it mean when they start to function, and what are these emergent properties that happen as they function together? And we talked about an example of ants. So you can zoom in and take a look at an ant or a couple of ants, and they may be running around in a bunch of different directions. But if you step back and take a look at how a whole colony of ants are working, you'll start to see these emerging behaviors. All the ants streaming in a particular direction or back and forth between a food source and their colony, oftentimes working together to bring food back to the colony or to help clear out debris from the colony, such as dragging a rock out of it. I'll stop right there.
Thanks. That's a great answer from that room. I also want to kind of tie it back into this response from room 3. Like you all mentioned the elemental cycles, room 3, in the last question, nitrogen and carbon, and this like we talk about like fertilizer added to agriculture to help plants grow, but can negatively impact the surrounding environment. something that comes to mind about, think about Haber-Bosch fertilizer, that liquid fertilizer where it's ammonium nitrate. That is an inorganic compound that doesn't occur naturally in the environment, and it actually can, in some instances, short circuit the microbial community's nitrogen cycling ability, because it starts to select for organisms that can grow very, very rapidly from ammonium, or doing the conversion of nitrate to other compounds. So super interesting process, and it's something to think about too, like that these decisions that are made throughout the agricultural process do have some systems level effects. Breakout room 3 says we're less likely to miss something if we think at the system level, reductionism versus the system approach. You might miss interactions if we study in isolation. Would somebody from room 3 like to expand on that? Because I could play devil's advocate, even though I'm presenting the systems biology side of things today.
So one of the like main things that we were talking about, like we kind of referenced like nutrition, and the idea of there being like, you know, however many nutrients that we need in our bodies, right? So if we're talking about like studying one nutrient in isolation, we might not be able to see, like, you know, as room 1 said, like the bigger picture, and also like you can study what could happen if the body is deficient in a nutrient, or if there's excess in a particular nutrient. But you might not see like how that interacts with the rest of the nutrients that like fuel the body. So in terms of in terms of ratios of certain nutrients, does that make sense?
Yeah,
so that's what we were getting at, really.
Yeah, so you're talking about like sort of the levels of different metabolites in an organism over time, and how one being very elevated might affect another. Okay, cool. That's a great.
Yeah. So that's just one example.
I want to push you to do your devil's advocate trick, right?
My devil's advocate thing is that it's really hard. It's really, really hard to do this, like at a systems level. It's hard to conceptualize everything that you could possibly think of. And I think that's where some of the advances in like artificial intelligence and neural networks come into hand, because they take into account uncertainty in a different way than like what we as scientists can do, like when we're designing experiments and trying to test these things. So it's like, there's also this element of we want to think about it as a system, but we also want to quantify how uncertain we are about the relationships to sort of ground truth our network and system level thinking. And this is how much we couldn't come up with explanations for. And it's like, that's the open-ended question. And that's why at the end I said it was like an iterative process, because there's always going to be that little tiny bit of like error rate, and that little tiny, like, you miss something, maybe somebody discovers some new RNA process that, you know, couldn't have been accounted for prior to the development of some unnamed molecular biology technique. So that's my devil's advocate.
And the reason I was pushing you to do that is I think there's a tendency to think like you were talking about, Shanine, in that note, reductionism versus system. And I think it's maybe a mistake to think of them as being adversarial. I think you need both. And I think it's important to keep your head wrapped around both.
Yeah, I agree with that, Cliff. Like when I talk to people and do some of these sort of network analyses for like plant biologists, it's like, you know, they have designed a discrete experiment where they're testing like a, you know, compound A versus compound B, and they're looking for an effect. But, you know, the GWAS, the genome-wide associated studies, only get you so far. And so it's like, you know, like, do you have enough information to do this? Like that sort of thing. And so that's kind of like where, you know, we've gone on this sort of story arc with this whole workshop series to this point where now it's like we're trying to come up with even more complex associations than we could possibly do with like a genome-wide association study alone. Does anybody have any like further questions or comments?
Excellent. Well, for those that I see people signing off, for those that do remain, just quick round of applause to both of our speakers today. And just to remind you that our final workshop is going to be next week. The final two topics is we're going to have Dr. Ellen Goddard talk about the ethical and social considerations as we move forward with genotype phenotype research. And then we're going to have a series of short talks from researchers in the G2P space, just to sort of highlight some of the wonder and some of the challenges. The idea of this is to help people sort of synthesize where we currently are and why we need to engage with broader interdisciplinary efforts to really solve a lot of these challenges. As Ryan mentioned, we have amazing ability to take a look inside of cells and see a lot of molecular actors or a bunch of individuals working. But we still have a lot of challenges in terms of, number one, putting them all together in order to see what phenotype or emergent behaviors they can create. And ultimately, and probably one of the more of the harder problems, is being able to develop predicting models to help predict what these things will look like, based either just on the genetics alone or taking a look at how some of these actors within cells and organisms function to create those phenotypes. And so with that, just a quick round of applause. Thank you, Ryan. Thank you, Cliff. And we all look forward to seeing everyone next week.